So taking a look at that unigriffin activator that you, how does DNA determine traits activator? And the reason, again, we are talking about this and we're having you do this is to give you one more opportunity for us to reinforce the ideas of DNA to mRNA called transcription, yep. And then this circle is an mRNA codon chart. So the 3 letters of mRNA code for an amino acid. Put a bunch of amino acids together, we get a protein. Proteins express traits. So that's what this example is trying to show you, that that amino acids code for a trait, and we kind of skip the protein step. But we are going to go through, and we are going to assume that we kind of jump ahead to the traits, and if we say, hey, here's the traits that you should have gotten, then your mRNA sequence got you the right amino acid sequence, which then gives you the right trait. And if the answer is you had the wrong trait, then we should kind of work up. But we're not going to go through letter by letter by letter because that gets a bit redundant, not redundant, a little bit more than most of you need. Okay? So the ideas of these, we go from DNA to mRNA through process of transcription, and then this 3-wheel mRNA sequence gives us our 3-letter abbreviations. So the traits that we had based upon the amino acid sequence says that it should have a tail like a horse, it should have brown fur, it should have no unicorn horn, and a lion's mane. And again, if you didn't have those traits, then you probably had the amino acid sequence wrong. And if you had the amino acid sequence wrong, you probably had one of two things. You read the chart wrong, or you had the wrong mRNA. You did the wrong transcription to mRNA. So take a look and try to figure out where you messed things up. The flow of genetic information, once again, is from DNA to mRNA. That's a process called transcription. From mRNA to tRNA is a process called translation. That tRNA, that 3-letter sequence of tRNA brings in a very specific amino acid. You take a bunch of amino acids together, we form an amino acid sequence. And that amino acid sequence then forms a very particular protein, and the proteins that we make express the traits that you have. If you change something along the way, if the DNA gets mutated, or the mRNA, or the tRNA, or the amino acid sequence change, then the protein changes, which then means your traits are going to change. Now this is all the in-between pieces. You know, a more general summary would be DNA to RNA to proteins to traits. You know, we talked about amino acids are like words of a sentence, like amino acids are part of a protein. Okay, so that's the idea. Now, your guided notes from the weekend talked about mutations, and in class last week we talked about mutations. There's 2 different types of mutations that we really focused on: a point mutation or a frame shift mutation. Generally speaking, which type of mutation, point mutation or frame shift, tends to cause more significant issues? Point mutation, where thank you. A frame shift is going to be more significant, right? So a point mutation is where a 3-letter codon is affected, which changes a amino acid, where a frame shift shifts every 3-letter codon after that. So generally speaking, a frame shift is more significant, leads to bigger implications or bigger issues. However, there are some types of point mutations that could be pretty significant. Imagine for a moment if the point mutation affected your start codon or your stop codon. That would be a significant mutation because you never started making a proton, or you didn't stop making that, protein, or you didn't stop making that protein where you should have. That would be a rather significant mutation at that point in time. Okay? So frame shifts tend to be frame shifts tend to be more significant when we talk about mutations. Any questions about this activity? Because on your test, there will be something very, very, very similar to this. Okay, so then, I know I handed a study guide to you guys as you were finishing up, but I would like you to pause from that, and we have a 9-minute video we want to show you. The other thing that we talked about in your section 4 guided notes was a very particular type of mutation called GMOs. So when we talk about GMO stands for genetically modified organism, and really what we called it, our kind of definition, we called it a man-made mutation. It's something that humans purposely mutated or changed the DNA of an object, the genetic information, for a particular reason. So any organism that has had their genetic information modified by a scientist is called a GMO. GMOs kind of get a bad rap. So this is a 9-minute video that kind of gives you some intro on GMOs, what they are, some examples of what GMOs are, and kind of what they do with them. So we're going to watch this. So like I said, I know I gave you a study guide. Please pause on the study guide and then watch this video. And we will give you some time to kind of wrap some of this other stuff up, some other study stuff. So here's the 9-minute video.
GMOs are one of the most controversial areas of science. Genetic engineering is used in many fields, but even though medical applications like GM insulin are widely accepted, the debate heats up when it comes to food and agriculture. Why is that? Why is the same thing treated so differently? Let's try to get to the bottom of this and explore the facts, the fears, and the future of GMOs. Humans have been genetically modifying plants and animals for thousands of years. Maybe a few of your crops had very good yields, maybe one of your wolves was especially loyal. So you did the smart thing and bred the plants and animals that had traits beneficial to you. Traits are just an expression of genes. So with each generation, those genes got more pronounced. After thousands of years, almost every single plant and animal around us is vastly different from its pre-domesticated state. If humans have been changing genes for millennia, what makes a so-called genetically modified organism, or GMO, different? Selective breeding is basically hoping for lucky hits. Genetic engineering eliminates this factor. We can choose the traits we want, make fruit grow bigger, immune to pests, and so on. So why are people concerned about them? Let's start with one of the most common objections to GMOs. Gene flow, meaning GM crops could mix with traditional crops and introduce unwanted new characteristics into them. There is a method that might guarantee complete prevention, but is a big anti-GMO argument by itself: terminator seeds. The idea is that they could produce sterile plants, requiring farmers to buy new seeds every year. The very concept of this, however, caused a public outcry, stopping the technology being put to use. This brings us back to the unintentional spreading of engineered DNA. There have been cases of GMOs growing where they weren't planted, and traces of modified genes found in foreign crops. But GM plants can't run wild entirely. Many crops pollinate themselves, and all crops have to be related to mingle. There are also cultural methods like buffer zones to keep unintentional crossing at a minimum. But if it's possible in principle that a GMO could unintentionally cross with a non-GMO, there's actually a more important question. Is food that comes from GM crops different to food from non-GM crops? This question has been a major concern from the very beginning. GM plants that are destined to be eaten are checked for possible dangers, and the results are evaluated by multiple agencies. After more than 30 years and thousands of studies, the science is in. Eating GMO plants is no more risky than their non-GMO equivalent. But don't just take our word for it. Sources for this and other claims are in the video description. But what about plants that have been engineered to be toxic? For example, Bt crops. A gene borrowed from the bacterium Bacillus thuringiensis lets engineered plants produce a protein that destroys the digestive system of specific insect pests. The plant makes its own pesticide. Insects that eat it die. That sounds alarming. Pesticide sprays can be washed off, while the poison in Bt crops is inside the plant. But actually, it's not a big deal. Poison is really just a question of different perspectives. What's harmless to one species might kill another. Coffee, for example, is a poison that kills insects but is harmless to us. Or take chocolate. It's dangerous for dogs, but a pleasure for humans. Bt crops produce a protein that is tailored to the specific design of the digestive tract of certain insects. It's completely harmless for us. There's also the opposite approach. Plants that are engineered to be resistant to certain weed killers. This way, farmers can use them widely, killing the other plants competing for resources without harming the crop. Here we get to the dark underbelly of GMOs. For the pesticide industry, they are big business. Over 90% of all cash crops in the US are herbicide resistant, mostly to glyphosate. As a result, the use of glyphosate has increased greatly. That isn't only bad. Glyphosate is much less harmful to humans than many other herbicides. Still, this means farmers have a strong incentive to rely on this one method only, casting more balanced ways of managing weeds aside. That's one of the most fundamental problems with the GMO debate. Much of the criticism of this technology is actually criticism of modern agriculture and the business practice of the huge corporations that control our food supply. This criticism is not only valid, it's also important. We need to change agriculture to a more sustainable model. GMOs as a technology are actually an ally and not an enemy in that fight, helping to save and protect nature and minimize our impact on the environment. Let's look at some positive examples. Eggplant is an important crop in Bangladesh, but often whole harvests are destroyed by pests. Farmers had to rely heavily on pesticides. Not only was this very expensive, farmers also frequently got sick. The introduction of a new GM eggplant in 2013 stopped this. The same Bt protein we talked about before, an effective killer of insects but harmless to humans, was engineered into them. This reduced insecticide use on eggplants by more than 80%. The health of farmers improved and their income rose dramatically. And sometimes a GM approach is the only option. In the 1990s, the papaya industry in Hawaii was under attack from the ring spot virus, which threatened to wipe out Hawaiian papaya. The solution was a papaya genetically modified to be vaccinated against the virus. Without it, the state's papaya industry would have collapsed. All these stories show a very narrow application. 99% of all GMOs we use right now produce pesticides or are resistant against them. There is so much more we could do. Scientists are working on GMOs that could improve our diet. Plants that produce more or different nutrients, like fruit with higher antioxidant levels that help to fight diseases, or rice with additional vitamins. On a larger scale, we're trying to engineer plants that are more resilient to climate change. Plants that can better adapt to erratic weather and adverse soil conditions, making them resistant to droughts or floods. GMOs could also not only reduce agriculture's impact on the environment, but actively help to protect it. Scientists are working on crops that can draw nitrogen from the air like microbes. Nitrogen is a common fertilizer, but its buildup pollutes the groundwater and speeds up climate change. Plants that collect their own nitrogen can fix two problems at once: the overuse of fertilizers in the developed world, as well as the shortage of it in developing countries. We could even modify plants to become super effective carbon collectors like the American chestnut tree, to mitigate and actually reverse climate change. With the tools we have today, our imagination is the limit. The world eats 11 million pounds of food every day. A UN estimate suggests we'll need 70% more by 2050. We could grow that food by clearing more and more forests to create fields and pastures and by using more pesticides. Or we find a way to do it on the land we've got right now with more effective methods like GM crops. Intensifying farming instead of expanding it means that GMOs could become the new organic. In a nutshell, GMOs have the potential to not only drastically change agriculture, but to also dampen the effects of our own irresponsible behavior. GMOs could be our most powerful weapon to save our biosphere. This video took more than 600 hours to make, which would be impossible without viewer support on patreon.com. If you'd like to support carefully researched content made with love, it's really very helpful, and you can get your own bird as a reward.
Okay, so GMOs, genetically modified organisms, they talked about I think it's glycophosphate was the one that most cash crops already have, are using, and I yelled Roundup, because maybe you'll hear that about Roundup Ready corn or Roundup Ready soybeans, and the idea of Roundup Ready is there is this strand of DNA that makes those crops immune to Roundup, which is just Roundup is a weed killer, right? You spray it on most plants and it kills most of the plants that it hits. So what they were able to do is instead of having going through and being very selective and weeding, you're like, hey, I plant a field of corn. You want that corn to grow. You don't want weeds competing for nutrients and water and sunlight. So if you can weed your garden, right, your crops are going to grow better. But what the farmers can do is they can just round up the entire field and it will kill everything but their corn or their soybeans. So that's one of the things they reference. So when we talk about genetically modified organisms, there's a lot of genetically modified organisms out there that we're looking at, we're using, and that kind of is a little bit of insight as to what it is, more than just like that one or two slides that was on your notes. I want to show you a few things in Canvas quick, and then we'll give you some work time. So on Wednesday or Thursday, if you're watching on video, maybe it's a Wednesday for you, but in class we're talking today, on Thursday you will be taking your test. So this is today's agenda. We did the activator, we watched that video. Now we're going to give you some work time on these last two things. One of which is and it's also linked under your virtual to-do list for tomorrow, is one of two things. One is a study guide, okay? There's your study guide on DNA and RNA, and there's a section for Quizlet tests. So we've done a Quizlet for section 1 and section 3. We've already done Quizlet tests for those on like your cold days or a snow day or a Thursday, Friday. We've already done section 1 and 3. They are linked here if you want to review those flashcards. They are linked under tomorrow's virtual day to-do list. But section 4 Quizlet test is the last Quizlet test for the last stack of flashcards for this. So those are I think we've done enough of those that you're pretty good. Some of you are new to us maybe, so our study guides that you got a paper copy of, you could go through. There's a Google Doc, there's also a PDF. One thing we do want to point out too is that if you get stuck or when you're done, you can check your answers because we have posted an answer key. Now, if you just blindly copy the answer key, that's not going to be good for your learning. The idea is, did you get did you get them right, or hey, I really got stuck on something and I wasn't sure what they were looking for. That's the intent of the answer key, but they are posted under the assignment for your study guide. So again, on the end of this week, we are testing on DNA and RNA, transcription, translation, amino acid sequence, proteins, traits, and that's what we're going to ask some questions about. So that study guide is a good review. We have some time. We are officially done at 11. Okay, so we have about 12-ish minutes left, 13-ish minutes left for you to get working on your study guide. Maybe you want to work on your section 4 Quizlet test, submit that screenshot when you're done. But now is a good opportunity for you to ask some questions and get help before the test that we have at the end of the week. So the rest of the time is yours to review, to help you prepare. If you have questions, please let us know.